The overall goal of the following experiment is to measure mitochondrial fusion in highly resolved 3D images of multiple cells in parallel. This is achieved by first expressing mitochondrial photoactivatable GFP in the cells of interest. Next, the GFP is photoactivated in a small region of the mitochondrial population in each cell. Then, 6 optical sections of each cell are collected every 15 minutes until signal equilibration is reached in order to assess the extent of mitochondrial fusion. Ultimately, a dilution of the mitochondria-targeted photoactivatable GFP intensity in cells undergoing mitochondrial fusion based on quantification of the fluorescent signal can be assessed.
The main advantage of this technique is that it is automated and therefore high-resolution data of multiple cells can be collected more quickly.
Though this method will be demonstrated using INS1 cells and provides insight into diabetes, it can be applied to other cell types to study the role of mitochondrial fusion in aging as well as metabolic and neurodegenerative disease.
Visual demonstration of this method is critical as the imaging automation steps are not described elsewhere.
After culturing INS1 cells to 80% confluence, detach the cultures with 0.05% trypsin and plate the resuspended cells onto poly-D-lysine-coated coverslip-bottomed imaging plates. After 2 days of culture, add mitochondrial matrix-targeted adenoviral photoactivatable GFP to the plates.